This is week 2, the first through the third week of human development, neural tube defects, and congenital heart defects, part 1. The learning objectives for this week are to describe the transport of gametes, the phases of fertilization, cleavage of zygote formation, and implantation of blastocyst, formation of amniotic cavity, and the chorionic sac. We will describe the process, gastrulation, neurulation, and formation of the neural tube. We will describe early and later development of the cardiovascular system and discuss the clinical presentation and timing of maldevelopment resulting in anencephalia and spina bifida. We will discuss environmental factors associated with neural tube defects. We'll describe fetal and neonatal circulation and discuss the embryologic origin and clinical presentation of congenital heart defects. This is a great slide showing a secondary oocyte and numerous sperm at the time of fertilization. And you can see how the sperm are all around the secondary oocyte. But what's also very important to look at is the fact that there's such a great discrepancy in the size of the sperm and the secondary oocyte. If we look at the oocyte at ovulation, we can see that during the process of ovulation, a secondary oocyte and the first polar body are released, and both are surrounded by the corona radiata. The oocyte is suspended in metaphase of meiotic division number 2. So in this slide, you can see the first polar body, the corona radiata, and the zona pellucida. In a previous slide, you saw the oocyte that was surrounded by all of the sperm. And as mentioned previously, if you look at the head of the sperm, there are a lot of acrosomal enzymes which are found there. And what they do is it helps to penetrate into the egg. A single sperm, however, will make contact with the oocyte membrane, and the membrane fusion occurs, triggering oocyte activation. And when this occurs, this is when you get the completion of meiosis. So you can see in this picture here, the fertilizing spermatozoon and the secondary, or the second polar body. Then the next thing that happens is that a pronucleus is formed. The sperm is absorbed into the cytoplasm of the oocyte, and the female pronucleus develops. And then the male pronucleus develops, and spindle fibers appear in preparation for the first cleavage division. In this slide, you can see that amphimixis occurs and cleavage begins. Amphimixis is actually when the male and the female pronuclei fuse. And then you can also see in this picture the metaphase of the first cleavage division. And now the first cleavage division is near completion, and this is about 30 hours after fertilization. And you form 2 blastomeres. And what the blastomeres are the cells that are produced after the cleavage of the zygote. The time spent in prenatal development is known as gestation, and it is divided up into 3 integrated trimesters, and each one of these lasts about 3 months. If we look at the first trimester, it's this time period in which we have the embryonic and early fetal development. And during this time, you can start to see the major organ systems appear. The second trimester is where we have the development of organs and organ systems, and the body shape and proportions change. In the third trimester, there's rapid fetal growth and deposition of adipose tissue. And during this time, most major organ systems are fully functional. In fact, an infant born 1 or even 2 months prematurely has a reasonable chance of survival today. So when looking at the first trimester, we can divide it up into 4 major stages: cleavage, implantation, placentation, and embryogenesis. The process of cleavage is a sequence of cell divisions that begins immediately after fertilization. The zygote becomes a preembryo, which develops into a multicellular blastocyst. And this part of the first trimester ends when the blastocyst contacts the uterine wall. The next part is implantation. This begins with the attachment of the blastocyst to the endometrium of the uterus, and this sets the stage for the formation of vital embryonic structures. Placentation occurs as the blood vessels form around the periphery of the blastocyst, and this allows the placenta to start to develop. The placenta is a complex organ, and it permits exchange between maternal and embryonic blood. It supports the fetus from its formation early in the first trimester until just after birth, when the placenta stops functioning and is expelled from the uterus. From that point on, the newborn is physically independent of the mother. We will go over fetal circulation this week, just to make sure that everybody remembers that from other biology classes that you have taken. Embryogenesis is the formation of the viable embryo, and it establishes the foundations for all major organ systems. The first trimester is the most dangerous period in prenatal life. 40% of conceptions produce embryos that survive past the first trimester. If we look at the process of cleavage and blastocyst formation, we can see that identical cells are produced by cleavage divisions, and these are called blastomeres. The stage called the morula occurs after 3 days of cleavage. And what it is the preembryo is a solid ball of cells resembling a mulberry. And during the process that these cleavage divisions are occurring, the morula, or the solid ball of cells, is making its way to the uterus, and it reaches the uterus on about the 4th day since fertilization. The blastomeres then form a blastocyst, a hollow ball with an inner cavity known as the blastocele. The blastomeres are no longer identical in size and shape. The outer layer of cells is called the trophoblast. As the word trophoblast implies, trophos means food and blast means precursor. Cells in this layer provide nutrients to the developing embryo. A second group of cells, the inner cell mass, lies clustered at one end of the blastocyst. These cells are exposed only to the blastocele and are insulated from contact with the outside uterine environment by the trophoblast. In time, the inner cell mass will form the embryo. The next 2 slides show you the cleavage and how the blastocyst forms, and the movement down the fallopian tubes to the uterus. This slide shows you the production of the morula. Here you see the early morula, the advanced morula by day 4, and by day 6 you have the blastocyst with the inner cell mass, the blastocele, and the trophoblast. And then by day 7 it is ready to begin implantation in the uterine wall. Implantation occurs 7 days after fertilization. The blastocyst attaches to the uterine lining or adheres to it. The trophoblast cells divide rapidly, and they create several layers. At the point of contact on the uterine wall, the trophoblast cells divide rapidly, making the trophoblast several layers thick. and the cells closest to the interior of the blastocyst remain intact, forming a layer of which is referred to as the cellular trophoblast or the cytotrophoblast. Near the endometrial wall, however, the plasma membrane separating the trophoblast cells disappear, and it creates a layer of cytoplasm. And this has multiple nuclei. This is at day 8. The outer layer is called the syncytial trophoblast, and it erodes the path through the uterine epithelium by secreting hyaluronidase. The next two slides show you days 6, 7, and then you'll see 8 and 9, and this process is showing you how the implantation process works. And this is the second part, which is the day 8 and 9. You can see the cellular trophoblast. You can start to see the amniotic cavity. In most cases, implantation occurs in the fundus or in the body of the uterus. However, in the case of an ectopic pregnancy, implantation occurs somewhere else. Ectopic means abnormal. And these other places could be in the abdomen or in the uterine tubes. Approximately 0.6% of pregnancies are ectopic pregnancies. They do not produce a viable embryo, and they can be life-threatening to the mother. As the capillary walls are destroyed in the uterus during the implantation process, maternal blood begins to come through the trophoblastic channels known as lacunae. And finger-like villi extend away from the trophoblast into the endometrium. They increase in size and complexity until around day 21. The inner cell mass has little apparent organization early in the blastocyst stage. However, by the time you get to implantation, the inner cell mass is separated from the trophoblast. The separation gradually increases, creating a fluid-filled chamber called the amniotic cavity. The trophoblast will later be separated from the amniotic cavity by layers of cells that originate at the inner cell mass and line the amniotic cavity. These layers form the amnion, a membrane that we're going to talk about later on. When the amniotic cavity first appears, the cells of the inner cell mass are organized into an oval sheet that is 2 layers thick. A superficial layer faces the amniotic cavity, and a deeper layer is exposed to the fluid contents of the blastocele. This slide is showing you the yolk sac formation, which is at day 10. And what's happening is that while the cells from the superficial layer of the inner cell mass migrate around the amniotic cavity, forming the amnion, the cells from the deeper layer migrate around the outer edges of the blastocele. This is the first step in the formation of what is called the yolk sac, a second extraembryonic membrane. For about the next 2 weeks, the yolk sac is the primary nutrient source for the inner cell mass. It absorbs and distributes nutrients released into the blastocele by the trophoblast. We're now up to day 12, and at day 12, superficial cells of the blastodisc have begun to migrate towards a central line known as the primitive streak. And at the primitive streak, the migrating cells leave the surface and move between the 2 existing layers. The movement creates 3 distinct embryonic layers: the ectoderm, consisting of the superficial cells that did not migrate into the interior of the blastodisc, the endoderm, consisting of cells that face the yolk sac, and the mesoderm, consisting of the poorly organized layer of migrating cells between the ectoderm and the endoderm. Collectively, these 3 layers are called germ layers, and the migration process is called gastrulation. Gastrulation produces an oval 3-layered sheet known as the embryonic disc. The disc will form the body of the embryo, whereas all the other cells of the blastocyst will be part of the extra embryonic membranes. If we look at the ectodermal contributions, we see that it's parts of the integumentary system, such as the epidermis, hair follicles and hair, um, sebaceous glands, sweat glands. In the skeletal system, some of the pharyngeal cartilages and their derivatives in the adult, and some of the other processes on bones are part of the ectodermal contributions. All the neural tissue, including brain and spinal cord of the nervous system, are from ectodermal contributions. If we look at the mesodermal contributions, we see that they consist of parts of the endocrine system, which consists of the adrenal cortex, the endocrine tissues of the heart, the kidneys, and the gonads, as well as all components of the cardiovascular system. Other mesodermal contributions are the integumentary system and the components of the dermis and the hypodermis, the skeletal system, all components except some pharyngeal derivatives, and all components of the muscular system. Some other mesodermal contributions are the lymphatic system, all components, the kidneys of the urinary system, including the nephrons and the initial portions of the collecting system, the gonads and the adjacent portions of the duct systems and the reproductive systems, and miscellaneous, such as the lining of the body cavities, the pleural, pericardial, and peritoneal, and the connective tissues that support all organ systems. Looking at the endodermal contributions, the thymus, thyroid gland, and pancreas of the endocrine system, the respiratory epithelium, and the associated mucus glands of the respiratory system, and the mucous epithelium, except the mouth and anus, the exocrine glands, except for the salivary glands, the liver, and the pancreas of the digestive system. And lastly, if we look at the endodermal contributions, we see that for the urinary system, the urinary bladder and the distal portions of the duct system, and in the reproductive system, the distal portions of the duct system and the stem cells that produce gametes. We now look at the formation of the extra embryonic membranes. Their function is to support embryonic and fetal development, and they consist of the yolk sac, the amnion, the allantois, and the chorion. Slide is showing the migration of the mesoderm around the inner surface of the cellular tophoblast to form the chorion. You can see the yolk sac, you can see the mesoderm and the blastocele, you can see the amnion, and you can see the syncytial trophoblast. And by week 3, the embryonic disc bulges into the amniotic cavity at the head fold. The allantois and endodermal extension is surrounded by the mesoderm and extends towards the trophoblast. You can also see the amniotic cavity, which contains the amniotic fluid, and the chorionic villi of the placenta. The yolk sac begins as a layer of cells spread out around the outer edges of the blastocele to form a complete pouch. It's an important site of blood cell formation. The amnion is a combination of mesoderm and ectoderm. The ectodermal layer enlarges and cells spread over the inner surface of the amniotic cavity. The mesodermal cells create an outer layer. It continues to enlarge through development. The amniotic fluid surrounds and cushions developing embryos or fetuses. The allantois is a sac of endoderm and mesoderm, and it gives rise to the urinary bladder. The chorion is a combination of the mesoderm and the trophoblast. The blood vessels develop within the mesoderm. The rapid transit system for nutrients that links the embryo with the trophoblast go through here in this first step in the creation of a functional placenta. The chorionic villi are in contact with the maternal tissues, and they create an intricate network within the endometrium, carrying the maternal blood. So if you want to do pre-implantation genetic diagnosis, it can be done 3 to 5 days after IVF of the oocyte. And what happens is 1 or 2 cells, the blastomeres, are removed from the embryo known to be at risk for a single gene defect or chromosomal anomaly. The cells are analyzed and then transferred into the uterus. You can determine the sex if taken from a 6 to 8 cell dividing zygote, and then this can be analyzed by PCR or FISH techniques. The polar body may also be tested for diseases where the mother is the carrier. We move on to talk about neural tube defects as well as congenital heart defects, and just wanted to point out that abnormal embryos and spontaneous abortions, we find that many zygotes, moruli, and blastocysts abort spontaneously. We know that they need adequate amounts of progesterone and estrogen to be supplied from the corpus luteum of the ovary. Remember, the corpus luteum is the shell that was left when the follicle released, the Graafian follicle released the egg at ovulation. And the corpus luteum is responsible for secreting progesterone and estrogen. And those two hormones are needed to ensure that the blastocyst can implant. Without a functional corpus luteum, you do not have the ability to have implantation take place. So we see that early spontaneous abortion rate is approximately 45%, and we know that another cause for spontaneous abortion is chromosome abnormalities such as the aneuploids that we looked at in the last course.